Anthoceros introduction. It is most widely distributed genus of class Anthoceratopsida of division Bryophyta having almost 200 species. About 25 species are found in Pakistan. The most common and worldwide distributed species is Anthoceros erectus. Habitat. The species of Anthoceros prefer to grow on damp soil, moist and shady rocks and other similar habitats. Some species grow on the decaying woods that found in moist shady places. Structure. External structure. The plant body is small, greasy, dark brown, dichotomously lobed, dorsiventral and prostrate thallus. The upper dorsal surface of thallus may be smooth, velvety or rough. The lowest ventral surface bears numerous unicellular smooth rhizoids. The thallus is attached to substratum by these smooth rhizoids. Tuberculate rhizoids and scales are absent. Bluish-green thickened areas on the ventral surface can be spotted called nostoc colonies. The thick middle portion is always without a definite midrib. Internal structure. The thallus of Anthoceros shows no internal differentiation of tissues. Therefore, it is not differentiated into storage region and assimilatory region. The thallus is composed of uniform, thin-walled, parenchymatous cells. Internal structure of Anthoceros thallus life cycle or reproduction. Or vegetative reproduction. It takes place by following methods. Fragmentation. The cells in the older portion die and eventually disorganize. The young lobes become separated and grow into new thallus by apical growth. Gamete. Gamete are known to develop on the dorsal surface of the thallus in some species of Anthoceros. They become detached from the parent thallus by the decay of connecting tissues and form new thallus. Tuber formation. The apices of thallus become thickened to form tubers at the end of growing season. These tubers remain dormant under unfavorable conditions and resume growth under suitable conditions. Or sexual reproduction. It is always of oogamous type. Species. Homothallic or heterothallic sex organs. Antheridium male, Archegonium female. Alternation of generation. Regular and heteromorphic. Gametophytic phase. So it is the dominant plant phase. Represents haploid generation and develops sex organs. Or the gametophytic phase begins as haploid spores are formed. Sperms and egg are the last structures developed during this phase. Sex organs are developed on the thallus lobes. In the monoecious or homothallic species, the antheridia are formed first and archegonia later. Gametogenesis. Antheridium. The antheridium is an elongated structure and has ovoid or pouch-like body developed in roofed cavities called antheridial chamber. The body of antheridium has an outer jacket layer of sterile cells called antheridial wall. This wall is protective in function. The antheridial wall encloses a mass of small fertile cubical cells called androcyte mother cells. Each androcyte mother cell has a denser cytoplasm and a larger nucleus. It divides to form two sperm cells known as androcytes. The protoplast of each androcyte metamorphosed into a single sperm. Each sperm is a minute, slender and curved structure having a pair of whiplash flagella at its anterior end. The sperms do not release from antheridium until enough moisture is present to permit them to swim over it. Archegonium. It is flask-shaped structure and consists of two parts. Venter. The basal swollen portion is called venter. It is directly attached to the tissues of thallus. It has a jacket of sterile cells called venter wall. It encloses the venter cavity which is filled with two cells, upper and smaller ventral canal cell, lower and larger egg cell, Neck. The long and slender portion is called neck. It consists of 4 to 6 neck canal cells surrounded by a layer of sterile cells called neck wall. When the archegonium reaches maturity, the neck canal cells and the ventral canal cells degenerate to form mucilage. The mucilage imbibes water and swells. A neck canal is formed through which sperm enter into venter that containing egg. Fertilization. It takes place in the presence of water. Upon maturation, the roof of antheridial chamber disintegrates to expose antheridia which absorb water and swell. The upper end of antheridial wall ruptures to release sperms. The mucilage oozes out from archegonia and attract male sperms chemotatically. Many sperms move towards egg but only one of them gain entrance into the venter through neck canal and fuse with egg to form zygote. The act of fertilization ends the gametophytic generation and is the beginning of sporophytic generation. Sporophytic phase. The sporophyte is complex and partially dependent on gametophyte. It consists of foot sater and capsule. The sporophytic phase represents diploid phase and begins as zygote is formed after the fusing of egg and sperm. The zygote increases in size and undergoes repeated cell divisions to form embryo. It almost completely fills up the venter of Archegonium. The walls of the venter divide pericolonally and form a two-layered calyptra around the young sporophyte. Structure of sporophyte: it consists of foot, stator and capsule. Foot: it is the basal, bulbous, embedded portion of sporophyte composed of parenchymatous tissues. It is deeply inserted in the tissues of thallus and form rhizoid-like structure into the thallus. It attaches the sporophyte with gametophyte and absorbs food from it. Stator: it is represented by intercalary meristematic zone and it continuously adds new cells at the base of sporophyte. As a result, sporophyte is elongated. Capsule: it is a long, narrow cylindrical structure tapering at apex. It shows three distinct regions: jacket, columella and archosporium. A. Jacket: it is 4 to 5 layered and present outside the sporogenous tissues. Its outermost layer is epidermis that contains stomata. B. Columella: it is a solid column of 4 to 16 rows of sterile cells in the center of capsule. Its functions including conduction of water and mechanical support. C. Archosporium: it is the fertile region of capsule present around the columella. It consists of sporogenous and sterile cell sporogenesis. The sporogenous cell divides to form sporocytes. The sporocytes are the last cells of sporophytic phase. Sporocytes undergo meiosis to form haploid spores. Spore is the first cell of gametophytic phase. These spores later on germinate to form gametophyte. Salient features of Anthoceros sporophyte: Anthoceros sporophyte exhibits some advanced features which are not found in other bryophytes. These features differ the Anthoceros from other bryophytes. These are following. The presence of photosynthetic tissues in the wall of capsule is step towards independency. The presence of columella in the center of capsule is step towards the evolution of vascular tissues. The presence of intercalary meristem at the base of capsule that continuously adds cell is step towards elongated sporophyte. The arrangement of archosporium in alternate sterile and fertile bands of cells a step towards the evolution of strobilus or cone. Rhizoids-like structures that grow out from the foot a step towards the evolution of roots.